So in previous lectures, you've learned about the process of cell division. And before cell division happens, during the S stage of interphase, the DNA is replicated. And that is done so that the new cells that are produced from cell division will each receive their own copy of the DNA molecules. So we are now going to look at that process of how the DNA is actually replicated. What does the process of copying a DNA molecule look like? So humans, right, have a lot of DNA, and that can be a little bit problematic sometimes with so many DNA molecules. So humans have what we call chromosomes, and chromosomes are basically DNA and proteins that have been condensed. They've been wound up into a very tight format that we recognize as a chromosome. And these chromosomes are visible under a microscope. You can actually see them, and it makes it a lot easier to divide out these chromosomes into the new daughter cells. So humans have 23 different chromosomes. So here we're showing a karyotype on the right-hand side, and it shows chromosomes 1 through 22. These are what we call autosomes. And then you have the sex chromosomes of either an X chromosome or a Y chromosome. So it's showing what's called a female karyotype, which shows all the chromosomes within that individual. And you see two X chromosomes for the female and an X and a Y chromosome for the male. But outside of that, they both have chromosomes 1 through 22. Chromosomes 1 and 2 are very large compared to chromosomes 21 and 22, which are a lot smaller in size. So each one of these chromosomes has to be copied during that interphase stage of S so that the new daughter cells that are produced will each receive a copy of those chromosomes. So keep in mind that along the length of the chromosome, there are different genes that can be found. So this is just showing kind of one chromosome, that of chromosome number 7, and it's showing in bands here different genes that can be seen. So we have an epidermal growth factor. Erythropoietin is a hormone for red blood cell production. And then we have a mutation. If there's a mutation in this area here, that would cause the genetic disorder of cystic fibrosis. So you can see that regions of the chromosome are associated with certain genes. And these genes carry the instructions on how to build different types of molecules. And that's what we'll be talking about in the next couple of little mini lectures, is how do we go from that gene to the actual production of a molecule. So these chromosomes, as I said, are all coiled up. And it's basically what we call chromatin that has been coiled into a chromosome. So over on the left at the top, it's showing a single chromatin fiber. So this is basically the DNA itself. You can't really see kind of the double helix. They're just representing it by a strand. And chromatin is actually DNA that is wrapped around histone proteins. Histones are a type of protein, and they're not really showing the proteins in this representation either. So technically, you have kind of a cluster of histones that are grouped together, and the DNA molecule kind of wraps itself around it, and then you might see another area where there's histone proteins that the DNA is wrapped around. All right. And so this makes what we call chromatin. So this is our chromatin fiber. So what happens during the S stage is that chromatin gets copied. So now we have a duplicate of that chromatin fiber, right? It's like we're taking it, we're copying it on a copy machine so that we have an exact replica of the original strand. Then we're going to wind up all of that chromatin. So we're just kind of winding it up on like this scaffold here. And as we wind it up, we're condensing it to make this chromosome. Now this chromosome is what we call a replicated or a duplicated chromosome, and it consists of the two copies, or what we call the sister chromatids. They're sisters of each other. Think of them as twins of each other. So this is one copy, and let me finish squiggling out everything here, and then let me change my color. So this is the other copy that has of chromatin that has been coiled up and condensed. So a lot of times we write a chromosome and we represent it like as the letter X, but really what we're showing by doing that is that it is actually 1/2 of our chromosome, our duplicated chromosome consisting of one sister chromatid, and here's our other sister chromatid, right? So one chromosome, and here's the sister chromatid or the copy of it, and it then makes that X-shaped structure. So a replicated chromosome consists of two sister chromatids. The sister chromatids are copies of each other, and we can condense those sister chromatids into a chromosome. And the chromosome, where the sister chromatids attach to each other or are connected to each other, is called the centromere. It's kind of the waistline of the chromosome. So now that we've got some of those terms out of the way, it seems like everything in this chapter starts with the letter C. Let's do a little bit of a review of our DNA molecule, since that's what's going to be copied. All right, so remember the chromatin consists of DNA wrapped around histone proteins. And now we're going to go back to just the DNA. Remember, DNA stands for deoxyribonucleic acid, right? So it belonged to that group of nucleic acids. So deoxyribonucleic acid. And nucleic acids are comprised of nucleotides. Remember, those were the building blocks of our nucleic acids. Now in DNA, the sugar that is used in each nucleotide is the deoxyribose part. So if you remember, a nucleotide consists of 3 parts: a phosphate group, a sugar, and a nitrogenous base. So we have our phosphate group, our sugar, and then we're going to add a nitrogenous bases. And at the time I said you need to know the names of the 4 bases that we find in DNA. You don't need to know their shapes, you don't need to know the shape still, but there's adenine, thymine, cytosine, and guanine. So therefore we can create 4 different nucleotides. One that will have a phosphate, a sugar, and a thymine attached, or a phosphate, sugar, and an adenine attached, a phosphate, sugar, and a guanine, or a phosphate, sugar, and a cytosine. So there's 4 different nucleotides that we can use as our building blocks to make up our DNA molecule. Our DNA is shaped as a double helix. So that means it has 2 nucleotide strands. So it has 2 strands of nucleotides that face each other and are then wrapped in that spiral pattern. So you see in this image, right, you see we've got our phosphate, our sugar, and then we have the nitrogenous base cytosine. Phosphate, sugar, and then adenine. Phosphate, sugar, and then thymine. So this is making up one of our nucleotide strands, and then across from it is our phosphate, sugar, guanine. Phosphate, sugar, thymine. Phosphate, sugar, adenine. So this is our second nucleotide strand. Now notice in the middle there are these dashed lines, and they represent hydrogen bonds that are holding our nitrogenous bases together. They're bonds between our nitrogenous bases. And that helps to hold these two nucleotide strands together. What you will notice is that adenine will always be across from thymine. Here we have thymine across from adenine, and guanine will always be across from cytosine. Here's cytosine across from guanine. So therefore, we write what's called our base pairing rules, and we say that the amount of adenine will always be equal to the amount of thymine, because on one nucleotide strand there's adenine, and we know across from it will always be thymine. Same thing, if there's thymine on one strand, adenine will always be across from it. So the amount of thymine should always be equal to the amount of adenine, or vice versa. Same thing with guanine and cytosine. If cytosine's on one strand, guanine will always be right across from it, or if it's guanine, cytosine will be across from it. So the amount of G will always equal the amount of C. So DNA replication, or copying the DNA molecule, the process by which this happens is what we call semiconservative replication. So semi-conservative. So semi, you think of half, and conservative is we're conserving something. So when we look at this process, if we start here with our DNA that has been kind of unwrapped, so it's not in a spiral pattern, and we still have our two nucleotide strands, and we can see that adenine's across from thymine and guanine's across from cytosine. What happens first is we go down the center and we break those hydrogen bonds between the nitrogenous bases. So in B here, it shows that the two strands have been pulled apart. Now we use these two strands as a template to build a new strand. So the dark blue is our old DNA as we move into C here, and the lighter blue are the new DNA strands that have been produced. So we can use the old strand as a template. It's a pattern that we use to build our new nucleotide strand, because we know if there's adenine on the old strand, across from it has to be thymine. If there's cytosine on the old strand, we need to place a guanine across from it, as well as our sugar and our phosphate that make up the rest of the nucleotide. So semi-conservative replication refers to the fact that the two DNA molecules that are produced, so here's one, here's our second one, right? That's the idea, is we want to make a copy of our DNA molecule, that the two molecules that are produced, they both consist of one old strand in dark blue and one new strand. One old strand and one new strand. So that is semi-conservative replication, where we're conserving the old DNA molecule, and each DNA molecule that is produced has half of that old molecule and half new.